Hi class, today we're going to finish the disinfection lab. We're going to do the absorbences after 24 hours of incubation at 37 degrees Celsius. Okay, so we've calibrated our spectrophotometer and balanced it out so that it reads 0. And then we have to just hit the mode button to go to absorbance. And then because the cells have been sitting for a while, they need to be spun out on a vortex. And so we'll just do that for a second, and then we can read them directly. Because we have writing on the tube here, we just need to make sure that the light goes through the side here. Okay, so we have an absorbance of the standard tube. That is our control that had no disinfectant. That is a 1.025. Okay, and then we're done with that one, so that comes out. And then we can go directly to our ethanol. And so our ethanol is here, so we'll spin it a little bit. And the writing's a little bit above, so we don't have to worry about that one. So it is 0.520. And then this is the 500 microliter of the ethanol added. This is going to be 0.115. So you can see it's starting to go down. And then here is even more, the 1,000 microliters. And then that will be basically 0.002. Okay, so that one's done. So we've seen a trend of downward trend for the ethanol. So the more concentrated, the better it is. And this is Virex. Virex is 0.960. And then for the 500, it is 1.52. And the 1,000 is going to be 0.002. And then the bleach, 250 microliters, is going to be 0.058. And then 500 is 0.141, and then 1,000 is going to be 0.210. Actually 0.266. I'm going to go back and check something here. The 500 one, make sure we vortex it well. And that's going to be 1.139, so that's close enough to our original one. And then I want to go back and check the Virex, because if you look at these three together, so we have the 250 on the left. Let's see if I can get this to show 250, and then 500, and then 1,000. So I need to vortex the 1,000 a little bit better. That is 1,000 of the Virex is going to be 1.4. Okay. Alright, so it was a matter of getting the material off the bottom. Alright, so then that is our absorbances. So our control, which was no disinfectant, was 1.025, and then ethanol with the 250 microliters 0.520, and then it went down 0.115 and 0.002 for 1,000, and then Virex is 0.96 for 250, 1.52 for 500, and 1.48 for 1,000, and then bleach is 0.058, 0.141, and 0.266. Now, because we had the Virex had some precipitate, we had these numbers to start with, so we had these are up there pretty well, and so we just need to remember that we have to subtract the original absorbances, because there's lots of clotting going on here, so this won't look so impressive after we do the subtractions. Okay, I think that's going to be it. So if you have questions, ask Christy, or you can email me, and that's going to be this lab. So all you have to do is take the information and put it on a calibration graph and figure out the CFUs per milliliter, and then you can go in and plot the graph for the CFUs per milliliter per concentration. So we can then determine which is the better disinfectant. All right, thank you. So I have the table here on this chart here that shows the different absorbances. So we have in the first column the agent used, ethanol is ETOH, and we have the 0 microliters is the control, and then 250 microliters, 500, and 1,000. And then at 24 hours I have the absorbance readings, and then at 0 hours we need to subtract those two. Subtract that from the 24-hour reading, and then that will be our difference. And so then all that is left for you guys to do is to use the calibration graph to figure out the CFUs per milliliter for each of those dilutions. Or each of those different doses of the disinfectant. And so, for example, you're going to take 1.014 and find that up on the left side of the graph, and then and then draw a line until it hits the dotted blue line, horizontal draw, and then you'll come straight down until you hit the x-axis, and then you'll have to do the interpolation to figure out the exact spot. But I just generally separate between the marks, so there's 122 and then there's 61 on the x-axis. So I just figure out what a fourth of those are. So I split into 4 areas along that, and just make a little bit easier to figure it out. So you can do that manually. So you can just print out this file, and I'll include it in the lab for Canvas, so you can have access to this to print it out, this PDF. All right, and then once that's done, you'll have this CFUs per milliliter for each condition and each concentration, and then you need to go in and do one more graph for finalize, or one more for each disinfectant, or you can do them all together on one graph. And so you're going to take and plot on the x-axis the microliters of each disinfectant, 0 microliters, 250 or 500 or 1000. And then on the y-axis, the vertical axis, you'll put the CFUs per milliliter. And so you can go from, you know, 0, about 5 to 122. And so then that'll be on the left axis. And I'll show that here, and then you can finish that one. All right, so your goal of this was to see which bacteria or which disinfectant kept the bacteria from growing, either by destroying it or inhibiting it. And you can see the trends of that, and which is the most effective disinfectant. Okay, thank you.